Hello students. Today we are going to continue with the second lecture of the lesson molecular basis of inheritance. Today we will start with the structure of DNA. Then we will move through RNA as genetic material and finally we will conclude with DNA replication. So let's study the first topic, which is the structure of DNA. The structure of DNA was given by Watson and Crick. As a matter of fact, DNA looks just like a twisted ladder. Twisted ladder, where the sides are composed of sugar and phosphate, and the steps are made up of nitrogen bases. The nitrogen bases are 4 in number, ATGC, adenine, thymine, and GC means guanine and cytosine. A and T, that means adenine and thymine, they are paired using 2 hydrogen bonds, while GC are paired using 3 hydrogen bonds. This complementary pairing helps the DNA to replicate accurately. Now what happens in some organisms? DNA is not present, it's absent. So here RNA acts as the genetic material. There are 3 types of RNA: mRNA, tRNA and rRNA. Messenger RNA or mRNA, it carries genetic message. Then tRNA or transfer RNA, it carries amino acids. And the third one is ribosomal RNA. As the name suggests, it forms rRNA. That means RNA forms ribosomes. rRNA, ribosomal RNA forms ribosomes. Now the third topic. What was the third topic? The replication, DNA replication. DNA replication is the process by which a copy of DNA is made. Before cell division, it is highly pertinent that DNA should make a copy of itself or should replicate, so the new cell gets the same genetic material. As a matter of fact, replication is semi-conservative. Here it means that the new DNA molecule gets one old strand and one new strand. And this was proved by Messelson and Stahl through his experiment. So for now, it was a very short video. Let's finish it off on high note, saying good night to all of you. Until we meet again, good night, have sweet dreams. Thank you, and don't forget to subscribe my channel and like my videos. Thank you.